In the early 80s, the dynamic compression plate was the most visible symbol of the AO group's accomplishment through a unique synergy of research, education, and industrial endeavors in the medical device field. But inevitably, changes were on the horizon. The dominance of plating in internal fixation was being challenged by the rapid acceptance of interlocking nailing. Even more importantly, several key experimental studies have shown that plating was causing disturbance of periosteal perfusion. Prior to that research, the sometimes dramatic loss of cortical bone beneath the plates had been fully attributed to stress shielding. However, this loss is now known to be mostly due to bone resorption as a natural response to bone necrosis. These findings defined the major goal for the development of the next generation plate: reduction of damage of periosteal perfusion. The first pilot project, called FIXIN, aimed to completely avoid disturbance of periosteal perfusion by eliminating contact between the plate and bone. The FIXIN project tested a plate application on intact sheep tibiae with bicortical screws locked to the plate by means of expandable slotted inserts. These experiments provided convincing evidence that keeping the plate a millimeter or two above the periosteum can practically eliminate disturbance of periosteal perfusion. However, the histology unexpectedly showed damage to the endosteal perfusion from the bicortical screws. Replacing bicortical screws with monocortical screws reduced damage to the endosteal perfusion. When locked, monocortical screws provide adequate load transfer from the plate to the bone. The project took on the new name of PC-FIX for a point contact fixator and began its decade-long testing in the lab, in experimental animals, and in clinical, veterinary, and human applications. Despite unusually positive outcomes in all the studies in every respect considered, including a human clinical study with close to 2,000 forearm fractures, the ultimate decision by the AO industrial partners was to ignore the demands of the AO to make PC-FIX available. In parallel with the technical developments aimed at solving the problem of iatrogenic damage to the bone, there was a major revision, or more appropriately, a revolution, in the surgical approach to internal fixation, which came to be known as biological osteosynthesis. If the mechanical goal of fracture stabilization is to be accomplished without plate-to-bone contact, the screws have to be locked to the plate. Locking the screws brings certain mechanical advantages, but the real goal is to preserve perfusion and thus reduce the consequences of bone necrosis, infection being the most consequential. The goal of preserving periosteal perfusion has been lost in the intervening years as locking plates gained acceptance in both human and veterinary surgery. The message from more than a decade of experimental work on hundreds of animals of keep the bone perfused has morphed into a much weaker one of lock the screws. As a consequence, most locking systems have not been designed to prevent iatrogenic damage to fractured bones. Alps offers strong fixation, but by design pays attention to bone biology. Alps 2, through its design, emphasizes bone perfusion. Alps 2 are cuttable and bendable. Fine-touch bone-holding forceps also complement the Alps 2 sets. Alps 2 has a low initial investment. Pictured below is a complete 6.5 instrument set. Pictured below is a basic 8-9 instrument set. Pictured below is a basic 10-11 instrument set. The full set for 6.5 includes the in-plane bending pliers. The basic sets for 8-9 and 10-11 do not include pliers. A universal set for in-plane bending for sizes 8 to 11 are currently available. KLS screw racks are made of titanium alloys, holding the screws with minimal contact, but safe enough to be handled in cleaning, sterilization, and transport.